today I'll be presenting the expression, purification, and characterization of 7249 protein. This was a project that I worked on with the MSKC group at Sarafan KMH in Stanford University, and my mentor was Dr. Daniel Fernandez. Oops. Okay, so the big picture here. So why is protein characterization important? Well, first I have to define what do I mean with characterization. So it's essentially understanding the protein. So what it binds to, what is the structure, how it behaves in specific conditions. And some of the applications that we may see with this technique is treatment of diseases, cell targeting, maybe for like cancer, or drug development, and then even like environmental purposes, such as like understanding like unique organisms or maybe pollutant degradation through enzymes. Okay, and then there you go. So just to provide a little bit more background, so the publication of Braula coeca or BLAU's genome has sparked interest in genes involving shared protein expression with its host, the Apis mellifera, also known as Western honeybee. So most of the research has been geared towards like immune system and metabolic pathways, but none of it has been geared towards like hydrolytic enzymes. And this is where we come in. And our purpose is to study and determine the structure of the hydrolase 7249 using recombinant DNA alongside with biochemical and molecular biology tools. So this is a methods overview, and I'll explain each of them a little bit more. So the first step is cell transformation. So we essentially take some E. coli cells and we add our plasmid with our protein DNA, and we get a transformed cell. And we hope that with these cells, we'll express our protein. So the next step is protein expression. Um, and essentially, we grow one of our colonies, as shown in the left, with multiple media. This is where we tested like multiple media and media conditions. And we essentially expand the growth of our cells. And then we collect those cells. And this is what they look like. They kind of look like ice cream. And each time that we collected those cells, we weighted just to compare the different conditions. And I'll talk more about that in the results section. So the next step was protein purification. And we essentially take those pellets and we add some lysis buffer. Then we sonicate them until the solution is homogeneous. And the solution kind of look like milk. And then we centrifuge that solution and we get a supernatant and a pellet. For the purification, we mostly care about the supernatant, and it gets passed through a machine called FPLC, and we are essentially able to separate some of the substances, and we collect them using some fractions, and we also get like this plot on the right that is essentially time on the x-axis versus concentration on the y-axis. And thanks to the fractions, we're able to do protein identification, which we can do through an AZS page, which essentially is just running a gel and see if the protein is expressed. And this leads me to the results section. So going back, this was like the first part, which was optimizing the culture growth. So these were all the media that we tested. And as you guys may see, the SB media with glucose had like the best average pellet weight per liter. And then you guys may also be noticing that there's a couple of them that don't have like bars. That's because the weights were unfortunately not recorded. And then there was also a couple of cell cultures that died, but that still gives us insight on what are the conditions that better support this cell growth. And then moving on to like the sort of identification part. So this was the gel we run. We used like 7 different lyses or 7 different FPLC runs, and they were all taken from the same fractions. And because we know like that our protein has a molecular weight of 65 kilodaltons, we know that in this red sort of box is where like we would find our protein, and we have some like lines there which indicate that our protein is indeed being expressed. However, we still have like some lines below, which means that there could be like some contaminant or um, that our protein is sort of breaking apart. We also concentrated our protein, and we were able to get about like 120 micrograms of protein, which was unfortunately not enough for crystallization. And crystallization is essentially the way that we would like model and get the structure of our protein. We would actually use like a technique that was previously mentioned by Alan and Tati, is the X-ray diffraction. And then just to summarize, with our data, we were able to find an optimal sort of medium, which is the SV with glucose. We were also able to identify our protein through the SDS page, and we also did a proteomic analysis with mass spectrometry, but I didn't show it for time purposes. And then for recrystallization, unfortunately, we were not able to get like a high enough yield for crystallize our protein and get like the actual structure. So next steps that we would like to take, there were still some remaining like cell pellets. So we would like to run those through the FPLC and see if they had any sort of protein. And then we just want to confirm and repeat the culture optimization. And then hopefully that would give us like enough yield for crystallization. So for my own reflections, okay, yeah, thank you. So for my own reflections, I learned that multiple biochemical and structural biology techniques. I learned to make and document observations in my notebook. I work independently on a laboratory setting, and most importantly, my main takeaway is that I gain confidence of being on a lab setting and lost fear of actually applying to internships. So with that, I just want to thank you, everyone, for being an amazing audience. And I want to thank all the people in this list. I had an amazing summer with this program, and I'm looking forward to what I'll do in the future.